My name is Manny Ascano, and I'm a professor at the departments of Biochemistry and Pathology, Microbiology, and Immunology. Yeah, so my lab is interested in looking at how cells respond to stress in the form of viruses or bacterial invasion or infection. And so with that, what I mean is that if you think of a virus is basically a package of genetic information that wants to copy itself. And it does that by infecting a cell and hijacking components of that cell. In order to do that, it's basically imposing its genetic will on a cell so that it can use the cellular components to basically make more copies of its protein and its DNA and RNA. And so my lab is particularly interested in looking at how DNA and RNA binding proteins, these are the proteins that actually can potentially interact with viruses, can detect that it is in fact a foreign RNA or a foreign DNA and be able to do that actually within an ocean of its own DNA and RNA. And it may seem like it's actually a trivial thing, but from a molecular perspective, these proteins need to be able to do this pretty fast. And so my lab is very focused on understanding these interactions and understanding whether this becomes an antiviral effect or a proviral effect. So one of the unique aspects of my work is that in many ways the viruses that we're looking at are always new. So with emerging infectious diseases, we're pretty much interested in things like chikungunya and Zika. These are obviously emerging viruses, and we're very interested in understanding how their basic ways of infecting a cell are either similar to what we already know or unique. And maybe in those situations where we find either similarities or uniqueness, these are opportunities for therapy, right? And so from a basic science perspective, we're trying to understand just the most basic interactions between host RNA or host protein and viral RNA. So my lab is a very interdisciplinary lab. We use tools within biochemistry, cell biology, proteomics, and now high-throughput genomics to try to understand, to basically bring to bear many types of assays and approaches to ask this question between, again, viral RNA and proteins. And so a main tool that we use in our laboratory is actually a photochemical biology tool where we are able to cross-link or permanently connect RNA to proteins in a way that we can identify either the RNA by high-throughput sequencing or the protein through mass spectrometry. Well, I think that this area of research in particular is an area where, if you think about it, a virus is constantly trying to infect a cell, and a cell is constantly trying to make sure that it detects a virus. So there's this constant push and pull and almost a molecular arms race, if you will. So this area is very ripe with unique biochemistry and unique mechanisms, and so that to me is always a good place to look at basic mechanisms of biology. Well, I think you can't separate basic science from biomedical research, right? And so again, with my field, when you think about what we're looking at here, it may seem almost academic just to think about viruses, but I don't think we would have ever predicted back in early days that if we were looking at chicken viruses, that this would actually usher in the idea of oncogenes and our modern understanding of cancer research, looking at chicken viruses. And more recently, looking at how bacteria actually use their own immune systems to be essentially combating bacterial viruses would end up becoming a new tool that we can use to edit the DNA of our own genomes. And these are all coming from very basic science, basic research, from viral research for that matter. So, yeah, I mean, my lab is fairly new, and so I've only been at Vanderbilt for 3 years. I've been a professor for clearly about as long as that, and so really, if I had to distill my successes, they are just a series of many firsts, right? So I would consider it a personal success that I was even able to go from one person, myself, in the lab, and be able to convince folks to buy into the science that I wanted to explore and get them into the lab and basically conduct the kind of work that we're doing now into an actual functional lab. So that's the first. About a year ago, we were able to receive our first federal grant from the NIH, and so that was a fantastic time, of course, for our laboratory, because I think this day and age, it can't be underestimated how difficult it has been for getting and landing research dollars for the NIH, and so it's been we have been very fortunate to be able to receive this federal funding. And then just recently, in fact just last week, we published our first paper in Nature Communications. And this was brought about actually by, so the very first person I hired was a research associate, and she happens to be the first author. And my first graduate student and my first summer undergraduate fellow are also co-authors in this paper. And so this just happened again last week. So, geez, you know what, I saw growth here, right? So since I've been at this, in this department, that's only been 3 years, there have been 5 new junior faculty here. So I saw a tremendous investment in the future. And that's just within our department. I think there's about 15 or 20 new junior faculty throughout Vanderbilt University. And so I consider this really an opportunity for myself to be in sort of a ground level of new growth in an environment that is being shared by multiple PIs at the same stage. And so I think this is a real opportunity for me. You know, trainees need to understand 2 concepts, right? That they need to learn how to fail fast and to be mentally agile. I think in the kind of work that we do, the experiments that we run often do end in failure, but with every failure there is always progress, and you can always interpret, appropriately so, some failed experiments and be able to see a path forward, even if that path forward is actually reversing. And so they need to understand that wholeheartedly and recognize that that is in fact progress, and to be able to be mentally agile enough, to be creative enough to move forward, right, and push a project. So I mean, that's, I think, that's where I would go, and certainly I wish I knew that early on. You know, in terms of that mental agility, I think part of the problem solving aspect of that is I would encourage students and trainees to go to seminars in fields that are not necessarily directly relevant to what they do, because I think, you know, and they need to be actively engaged in listening to these folks, not necessarily to learn what they're doing per se, but also try to understand how that they can use what they're hearing in their own research, you know. And I think this is a good idea to really recognize that creativity isn't really creating from nothing, but rather oftentimes it's more often that you find that you're basically borrowing established approaches in one area of biology and then just reapplying it in another in that creative sense. I was an undergraduate at the University of Illinois at Urbana-Champaign. I majored in microbiology and chemistry, minor in chemistry, and I worked with Ralph Wolf, who is one of the pioneering biochemists who looked at archaebacteria when archaebacteria didn't even have a domain name. These are a type of bacteria that is evolutionarily closer to eukaryotes, us, versus your normal bacteria. And so very early on I was very interested in this, again, a unique type of biochemistry. And from there I moved on to University of Cincinnati as a graduate student in the Department of Molecular Genetics, Biochemistry, and Microbiology, where I studied actually signal transduction in Drosophila melanogaster, so hedgehog signal transduction. There I learned a lot of things about biochemistry again, but this time more of a slant towards genetics and Drosophila and fly genetics. And you know, my love of signal transduction is sort of brought to the fore when I came to Rockefeller University in New York, where I started getting interested in the idea that RNA itself can be acted on as a substrate by proteins in a manner in which it could be dynamically regulated. And from there I'm bringing my training in signal transduction and now RNA biology into what I do now here in the realm of innate immunity. This space here that my laboratory is in is state-of-the-art. When I first moved here, actually this space was quite dated. I think it was the 1970s or 80s when it was last renovated, and so within this realm of wanting to invest in the future, they obviously offered me space, which then I was able to be a part of in helping the design and getting this actual lab into what you see now. And it's an open lab concept. There are approximately 5 or 6 laboratories that can fit in this space, of which I am one. And it sort of fosters a lot of collaboration because we're quite literally next to each other's laboratories. And in fact, the PIs, the professors that are in this space, are for the most part all junior faculty. So again, here is a situation where we're all starting in the same space, in the same place, and doing, you know, fantastic new science together.